Hello, my name is Mathew Fischbach. I'm a PhD student at the University of Minnesota's Bioinformatics and Computational Biology graduate program, and today I will be talking about my research using pathway-based genetic interactions to predict disease phenotypes with machine learning. Of the many discovered risk loci from genome-wide association studies, we still cannot fully explain the heritability of many complex polygenic diseases with these known loci. It's thought that the genetic interactions could be part of this missing heritability. And in the lab of Chad Myers, we've recently developed a GWAS method called bridging gene sets with epistasis, BRIDGE, and we have successfully discovered novel loci for a variety of diseases using genetic interactions. It's been shown that with BRIDGE, these genetic interactions tend to cluster into larger and structured networks that are spanning between pathway models and within pathway models, which I'll explain a little bit more about in a moment. My method is using a hierarchical pathway-based approach for predicting disease status. Using GWAS data genotypes and gene set pathway information, we can map the SNPs to the pathway layer. And in the pathway layer, each individual pathway will have one machine learning model that trains and then outputs from a decision function as features into a final phenotype layer, which takes all the pathways as features to then create a final phenotypical prediction. So, and here's a little bit more about the WPMs and BPMs like I promised. Within pathway models, WPMs only look at the genetic interactions within a single pathway, while BPMs look at the genetic interactions in between two different pathways. We can create genetic interactions and represent them three different ways as single, pairwise, and combined SNP types. Single SNP types are just using the number of risk alleles that an individual has for each SNP. And for WPM, pairwise genetic interactions come from the pairwise combinatorial matrix that results in symmetric matrix. So we only use the lower left triangle of that for our pairwise interactions. For combined, we would use that lower left triangle as well as the diagonal, which corresponds back to the single SNPs and combines the two, single and pairwise, into the combined. For pairwise, we can just use the entire matrix for pairwise combination and also use the SNPs from single and pairwise all as a feature set for a pathway. The data that we are using are two Parkinson's disease cohorts, but today we'll just be talking about the National Institute of Neurological Disorders and Strokes, the NINDS cohort. We are using canonical pathways from Molecular Signatures database, and after our pathway filtering, we are left with 685 pathways, which then corresponds to being able to have 685 WPMs within pathways. If we were to generate BPMs from 685 pathways in a pairwise manner, that would be over 200,000 BPMs that we could generate. Now, that's too computationally massive, so we use the significantly identified pathways from a bridge analysis of each cohort in order to generate our BPMs. For the NINDS cohort, that's 1,653 BPMs. We then use scikit-learn's logistic regression with lasso regularization for each pathway level WPM and BPM, and we shuffle that training testing split 10 times to average out the results. Here are the top 10 pathways identified from that pathway layer for the NINDS cohort. Notably, they are all dominated by BPMs, of which the BioCarta HIV NEF pathway is in every single one for the top 10. Looking next at the box plot, this is of all the pathways, all Ws and BPMs that we used. And on average, BPM does have a better AU-ROC than WPMs when we look at single, pairwise, and combined SNP interactions. If we just look at those SNP interactions just within a single pathway model, BPM or WPM, it's kind of hard to see any difference with this figure. So we go down to the ROC-AUC figures down below, and we compare the best performing BPM from the rank 1 against the best WPM, which was rank 11, and unsurprisingly, the best WPM was also BioCarta HIV-NEF pathway. These ROC-AUC plots show that for WPMs, there's not much benefit in using these genetic interactions as pairwise or combined SNPs. However, with BPMs, we do tend to see a benefit when introducing these genetic interactions, with an increase in that AUROC. Then feeding in all those pathways into our phenotype layer, we have the first plot up top, which uses all the pathways, and we see that our machine learning models cannot perform better than the average top 5 WPMs or average top 5 BPMs. So instead, we looked at the pathways and only took those that had a greater than 0.5 AUROC on average. This indicates that a pathway is performing better than random at predictions. And when we did so, we reduced our number of pathways from 2,338 to 1,799, and we see a dramatic increase in the average AUROC for all models used. Specifically, with the support vector classifier, we see that pairwise and combined both perform consistently better than their single counterpart. So this is suggesting that the use of these genetic interactions, BPMs, pairwise, or combined, improves overall phenotype predictions from genotypes. And in conclusion, we proposed here a method to predict PD phenotypes from GWAS genotype data using pathway-based genetic interactions, and have found genetic interactions to increase our ability to predict phenotypes. Thank you very much.